Paper 6, November 22, Variant 3. A student investigated the effect of concentration of glucose on the rate of respiration. أنا عايزة أشوف الإفكت بتاع concentration of glucose على rate of respiration. يعني this is my changed variable. This is what I'm going to change. This is my independent variable. Okay? Independent variable. The equation for aerobic respiration is glucose plus oxygen will give me water and carbon dioxide. The student uses this method. Label one test tube A and another test tube B. Third test tube is water. Add 1 cubic centimeter of 0.1 mole glucose to A. 4 cubic centimeter of distilled water to A. يبقى I put 1 glucose and 4 water. 1 glucose and 4 water. Use this information to calculate the concentration. How do I calculate the concentration here? This question can be repeated. So you need to know how to calculate the concentration. He said that he used 1 cubic centimeter from 0.1 mole. And he added 4 water, so he diluted it. I am going to put the volume of sugar solution used over the total volume. How much sugar did you use? 1 over 5. 1 over 5. Of which concentration? This concentration times 0.1 mole. Then you calculate it on the calculator. It will be 0.02. Again, volume of sugar solution used over the total volume times the concentration used. You can write it. Volume of sugar solution used over total volume multiply by concentration used. Use a glass rod to stir the yeast provided in the beaker. We said that we always stir, we still to distribute the contents and avoid settling down. Use a syringe to put 10 cubic centimeter of yeast in a glass rod to mix the contents. Add warm water to the water bath. Measure the temperature of the water in the water bath. Record the starting temperature. Of course, this will be an error. Any water bath can give me an error because it is not maintained. Pour water into the test tube called water and set up the apparatus as shown. This is test tube A containing the yeast and the glucose, and this is the water bath. Here is another test tube with water in it, and bubbles of gas will appear here. Bubbles of gas will appear. طيب, once the apparatus is set up, leave it for 2 minutes. Bubbles will start to appear. Count the number of bubbles in 3 minutes. This is another error. Counting the number of bubbles is not accurate. Remove test tube A. خلاص, after I finished, I will remove A, place it in the beaker called waste, and then I will put 5 cubic centimeter from 0.1 mole. But this is my concentration. I didn't add water. I just added this glucose solution. Stir the yeast suspension. Put 10 cubic centimeter of yeast. But this is a constant variable. Constant. Place test tube B in a water bath. Connect the bung, the delivery tube. Repeat steps and 8. You will count the number of bubbles. Measure the temperature of the water bath. Again, record the final temperature. Starting temperature, 35 degrees Celsius. Final temperature, 23 degrees Celsius. يبقى it decreased by 12 degrees Celsius. He recorded the number of bubbles in 3 minutes, drawing a line for each bubble. This is a tally count. 1, 2, 3, 4, 5, 5, 10, 12 bubbles, 5, 10, 15, 20, 25, 28 bubbles. Why is this? Because in test tube A, the concentration of glucose was 0.02, while in test tube B, the concentration was 0.1. More glucose, more respiration, more bubbles. Okay. Draw your table, prepare a table, record your results. Test tube A, as we said, what did we change here? We changed the concentration of glucose. Here it was 0.02, here it was 0.1. 0.02, here it was 0.1. But what is the number of bubbles? Here it was 12 and here it was 28 bubbles. Of course, the changed variable is on the left side of the table. Results or your dependent variable is on the right. Using the results, calculate the rate per minute, bubbles per minute. Now we said that he counted the bubbles in 3 minutes. So if I want to get the rate per minute, I will divide 12 by 3. It will be 4 bubbles per minute. 28 by 3, it will be 9.83 bubbles per minute. What is the conclusion? As the concentration of glucose increases, the yeast respires at a higher rate. This is why it gave you more bubbles. What is the variable changed? Independent. I changed the concentration of glucose and I marked it at the beginning. I want to see the effect of glucose concentration. But this is my independent variable. Independent variable will be the concentration of glucose. Why the temperature is a source of error? It is not maintained. Temperature is not maintained. What is an improvement for this error? Use thermostatically controlled water bath. Explanation and improvement. Why is it a source of error? Because it is not maintained, so it decreases with time. The temperature will decrease to reach the room temperature. What is my improvement? Using thermostatically controlled water bath. Why the yeast suspension was stirred in step 4 and step 12? I just told you, we do the stirring to evenly distribute, okay? Amount of yeast cells are the same in both test tubes. I'm doing the stirring to evenly distribute the yeast over all the test tube. And to avoid settling down. I do not want it to settle down. Why the test tube was left in water bath for 2 minutes in step 8? Let's check that. Step 8. Leave it for 2 minutes. Bubbles will start to appear. After 2 minutes, you will put the stop clock and start counting. طب why did I leave it 2 minutes في الأول? You have to leave it before you start counting to equilibrate or to reach the same temperature before you start counting. Equilibrate the temperature or to reach the same temperature of the water bath. Okay. A student wanted to measure the volume of gas instead of counting the number of bubbles. Draw and label the apparatus. Volume of gas is done using the gas syringe. So you will draw a syringe with graduation, and this is the plunger, and the gas will be inside the syringe. Okay? Type. Question 2. He started with plan and investigation. Actually, this exam had 3 questions. Plan an investigation to find out how the volume of water added to the germinating seeds affects the rate of germination. I'm going to follow my steps. Use 5 petri dishes, okay? Uh, put different volumes of water in each dish. Put the same number of seeds, the constant variable, same number of seeds, same type of seeds, same temperature and oxygen concentration. Okay? Measure the time taken for the seeds to germinate. You're going to put your dependent variable. How are you going to know which one is the best? Either measure the time, see which one germinates first, or you can measure the number of seeds germinated after a certain time. يعني after مثلا 2 days, count the number of seeds germinated in each dish. Maintain the air temperature of the room. Repeat at least 2 times and calculate average. As we said, you have to put them in the same temperature, either by putting them in a water bath, the petri dishes, or fixing the temperature of the room that they are in. The last question, question 3. Photograph of an in-cap mushroom. He wants you to draw it. Drawing the cap, mushroom cap, okay? I want to put, this is my drawing, this triangular structure. The stem is not smooth. The stem is not smooth. يعني it's a bit rough كده. Okay? And the mushroom cap, white cap on top. يعني separate white patches كده. Of course, you will not have to draw it all white patches. Okay? And it gets smaller as you go down. One mark was for clear continuous lines, no shading. I did not color anything. The other mark is that it's a big drawing, okay? Third mark, white patch across the top of the cap here, white patch here, and the cap drawn separate from the stalk. يعني this part is separate from this part. It's not continuous. You have to draw this with a straight line here. Number 2, either you can, the stalk, it has to be, the fourth mark يعني, the stalk has to be a rough كده, rough edge. It has to be rough edge. Or you can draw texture here برضو, it was accepted. اللي هو zigzag lines this. Okay? The white patches he didn't give away a mark for, but the most important is the top white patch. Even if you draw it, okay. After the drawing, he gave me a magnification question. Measure the length of AB using your ruler. Convert it to millimeter. I will multiply it by 10. It will be 65 millimeter. Use your measurement to calculate the actual width. Magnification equals image over actual. So I want the image over the magnification. Here is the magnification, 1.5. 65 over 1.5. Careful when you put the number, he wants it to the nearest 2 significant figures. This will give me 43.333. 2 significant figures, it will be 43. Numbers may change due to different scales of printing. In a study, the color of the cap growing in different temperature was recorded. As the color goes darker, he gives it a bigger score. This is the color score and this is the temperature. Hotter temperature, lighter color. طيب Plot a line graph, normal line graph. Left side of the table is on the x-axis, right side on the y-axis, cap color school. 15, 17, 19, 21, 23. This is going every 2 degrees Celsius, so you can put the numbers 15, 17, 19, 20, 23. Here I put it 0, 2, 4, 6, 8. I plot my points and then join point to point using a ruler. What is the information? Use information to describe the results. As the temperature increases, the cap color gets lighter because the score decreases. No change between 15 and 17. The other mark here, between 15 and 17, it remained the same. It remained dark as it is. The cap of the mushroom shown, figure 3.3 shows the cap of the mushroom shown grown at a constant temperature. Use the scale and your graph to estimate the temperature that this mushroom was grown. What score do I give this mushroom? What score do I give this mushroom? I think it would be مثلا a 2 or a 3. يعني if you choose 2, it's correct. If you choose 3, it's correct. Let's say I will give it score 2. طب what temperature is it grown? Let's see, score 2 here. And then I will go down. It is right in the middle between 23 and 25. So it will be around 24 degrees Celsius. You have to show the lines on your graph, how you got the number. Suggest why your answer is only an estimate, because anything that has colors, you cannot judge color by the eye. It is subjective. Just as we said, some people may give it score 2, some people may give it score 3. All of the mushrooms used were the same species. Why are they the same species? Because I'm only changing the temperature. And because if I use different species, they will have different colors. يعني I'm not changing multiple variables at the same time. I only change the temperature. Okay? One variable other than the species that should be kept constant. I'm changing the temperature. Any other thing should be constant. The amount of water, pH of the soil, light, nutrients in the soil, humidity. Anything else that affects the growth of the fungus should be the same. The last question, it contains vitamin C. Describe a method that you would use to test a sample of mushrooms for vitamin C. I will get the mushroom, crush and grind in order to create a liquid from it and extract. Add water. And then I will add DCPIP. DCPIP will change from blue to colorless if the vitamin C is present. Add DCPIP, it will change from blue to colorless.